Hello students, here we would continue with the topic molecular mechanisms of drug action. In my last session, I discussed about enzyme-linked receptors. Here I would explain about the intracellular receptors, ion channel receptors and G protein receptors with examples. Signaling mechanism in case of intracellular receptors. The intracellular receptors are located inside the plasma membrane. Hence, the substances or ligands that need to bind to these receptors should be able to move across the lipid bilayers, as shown in the adjacent figure. Example of such lipid-soluble substances are steroid hormones, vitamins, small molecules such as nitric oxide and hydrogen peroxide. As an example for signaling mechanism in intracellular receptor, we'll go with the glucocorticoid receptor. These receptors are placed within the cytoplasm. In the inactive state, when there is no binding of ligand, the receptors are bound to heat shock protein 90. Because of this, the DNA binding domain is masked or concealed as shown in the adjacent figure. When administered with glucocorticoid molecule, these molecules are lipid soluble and hence can pass through the plasma membrane. They bind to the specific receptors. The binding of the ligand to the receptor leads to the release of heat shock protein 90 and exposing the nuclear binding domain. The receptors are then translocated to the nucleus where they bind to specific steroid response elements on the DNA. This leads to upregulation of anti-inflammatory genes and downregulation of pro-inflammatory cytokinins. Gene regulation by steroidal receptors are slow in onset, but the response is long-term. Hence, it has therapeutical consequences. Firstly, the glucocorticoid molecules cannot be used for treatments to relieve the symptoms immediately. Example, glucocorticoid symptoms cannot relieve the symptoms of bronchial asthma immediately. All these drugs have a lag period of 30 minutes to few hours. Secondly, the effect, which could be beneficial or toxic, it cause persists for hours or days after the drug is stopped. Next, signaling mechanism in a ligand-gated transmembrane ion channels. I had discussed about this receptor in one of my previous sessions. Here, as an example, I would explain the nicotinic acetylcholine receptor. These receptors are best studied as they are evenly distributed throughout the skeletal system. The receptors is composed of 5 subunits, 2 alpha subunits and 1 each of beta, gamma and delta as shown in the adjacent figure. The acetylcholine transmitter binds to the alpha subunit leading to the opening of sodium channel. Sodium ions enter into the cell while potassium ions leave the cell, creating a localized excitatory postsynaptic potential depolarization. When this signal reaches the T tubules, it triggers the opening of another gated calcium ion channel which leads to muscle contraction. The time lapse between the binding of the ligand to the receptor and the response is in milliseconds. I will discuss about this in detail in the following slides. Communication between nerve and muscle occurs through neurotransmitters. An action potential at the terminus of presynaptic motor neuron leads to opening of voltage-gated calcium ion channels. An influx of calcium ion occurs. Neuron action potential leads to release of neurotransmitters acetylcholine from the synaptic terminal into the synaptic cleft. The acetylcholine molecules diffuse across the synaptic cleft and bind to the acetylcholine receptors on the motor plate. The binding of the acetylcholine to the receptors open the sodium ion channels. Sodium enters the muscle cells and potassium ion leave the muscle cells. This leads to a change in voltage, that is action potential. The action potential then travels across the membrane, reaches the T-tubules, and it triggers the opening of voltage-gated calcium ion, which is released from the sarcoplasmic reticulum into the cytosol. This leads to muscle contraction, which we would study in the next slide. The release of calcium ion from the sarcoplasmic reticulum into the cytosol leads to increase in concentration of calcium ion. The calcium ion binds to the calmodulin molecule, which activate myosin light chain kinase by phosphorylation and increase the myosin ATPase activity. The active myosin cross bridges slide along the actin and brings about muscle contraction. The acetylcholine esterase that is present at the synaptic cleft removes acetylcholine from the synaptic cleft by breaking it down into acetyl and choline. This leads to transport of calcium ion back into the sarcoplasmic reticulum. This leads to relaxation of the muscles, as shown in the second figure. The voltage-gated ion channels do not bind to neurotransmitters directly but are controlled by membrane potential. Hence, such channels are also important drug targets. Here, I have enlisted certain drugs which are used as inhibitors or mimic acetylcholine and certain modulators of ion channels. The next class of receptor is the G protein receptor and second messengers. The structure of G protein receptors was discussed in detail while teaching types of receptors. In this type of receptor, the signaling mechanism involves a class of protein called G proteins, which get activated when the ligand gets bind to the receptor. Activation of G protein involves binding of GTP to the alpha subunit and release of bound GDP. The alpha subunit dissociate from the trimeric complex and interact with the effector system to mediate specific responses. This changes the concentration of intracellular messengers like cyclic monophosphate, calcium, etc. Different subunits of G protein are associated with release of different secondary messengers. Example, the alpha subunit leads to stimulation of CAMP as seen in the figure above, while beta and delta subunits activate potassium channels. In the present session, I would like to talk about or discuss in detail the muscarinic receptor. The muscarinic receptor belongs to the class A rhodopsin-like receptor of G proteins. There are 5 types of muscarinic receptors: M1, M2, M3, M4 and M5. M1, M3 and M5 are excitatory receptors and stimulates phospholipase C, which then activate IP3 and DAG. The occurrence of this type is in the central nervous system, smooth muscles and endocrine glands. The M2 and M4 types are inhibitory receptors. They inhibit adenyl cyclase. The M2 type receptors are found in the heart, which is responsible for inhibition or slowdown of myocardial contractions. Signaling mechanism of M2 type muscarinic acetylcholine receptors. The binding of the acetylcholine to the receptor activates the G protein. It is catalyzed by the exchange of GTP to GDP on the alpha subunit. Here, the beta and the gamma subunit binds to the K channel. Remember, it is beta and gamma subunit. It increases the permeability of potassium ions, leading to hyperpolarization of the membrane. This results in reducing the frequency of heart muscle contraction. Activation is terminated when GTP bound to the alpha subunit is hydrolyzed to GDP and the alpha subunit binds to the beta and gamma subunit of the G protein. This is an example of inhibitory G protein signaling mechanism. Muscarinic acetylcholine receptor M1 type. These are excitatory in function. The binding of acetylcholine to the muscarinic receptor activates the G protein. The activated alpha domain binds to the phospholipase C. This leads to hydrolysis of PIP2 in the membrane, yielding two secondary messengers IP3 and diacylglycerol. DAG activates PKC, which can further lead to activation of MAP kinase cascades. The IP3 diffuse through the endoplasmic reticulum and interact with IP3 receptors, leading to opening of calcium ion channels. This releases the calcium ions from the intracellular storage sites that are selectively used to activate muscarinic receptors are atropine and scopolamine. These are the drugs that are used to dry up secretions and dilate the bronchi during anesthesia and to dilate the pupil during ophthalmological procedures. Antagonists of muscarinic acetylcholine receptors were the first to be used for treatment of Parkinson's disease. Certain drugs show selectivity in response based upon different cells. Example to this is tamoxifen, which acts as an antagonist on estrogen receptors expressed in mammillary tissue but as an agonist on estrogen receptors in bone. This is useful not only in treatment of breast cancer but also in prevention of osteoporosis. The table here enlists certain G proteins, their receptor and effector molecules. The present slide summarizes the different types of receptors and signaling mechanism. Thank you.